Well, thank you so much for having me and for the invitation today to give this webinar. Um, for this webinar, I will be talking about a publication of ours that came out last year, October, which is a meta-analysis about the impact of neonicotinoid on different aspects of bird life, actually. So I'm Elke. I am currently conducting a PhD at the Behavioral Ecology Group in Wageningen University, the Netherlands, which is actually about a different topic, also involving birds, but focusing on behavioral thermoregulation in Eswatini. But for today, as I said, we're going to go and talk about a meta-analysis. And we're actually going to start by talking about a different pesticide first, one of whom I hope most of you are acquainted with, which is DDT, a very famous and notorious pesticide that was in use mostly about 80 years ago after the Second World War, and used quite extensively across the planet, actually. And it was only until Silent Spring, written by Rachel Carson, actually arising or notifying us on the silence of the absence of bird singing in spring in the States, when the views on yay, DDT is good for me, kind of switched to nay, not really, actually. So it was first believed, actually, that this pesticide wasn't so harmful. It turned out later on that we were observing quite some disastrous and dangerous effects on many birds and animals where this pesticide was applied. And this has actually happened in a bit of a similar fashion now with this group of neonics that I'm talking about today. Where in the recent decade, and a little bit more than that, this group of pesticides has been starting to be associated with bird population declines. So we are definitely not the first to publish on this topic, but in a way we wanted to contribute to also quantitatively address the direct effects in this meta-analysis. So all around the globe nowadays, these neonics have been assessed, and also for many different non-target organisms. So not only birds, but also aquatic arthropods, as well as bees and butterflies, for example, because the suspicion was that all of these non-target organisms were actually affected, although non-intendedly so. So these papers served as inspiration, actually, for the current paper. So to briefly touch upon what these neonics actually are, they are a group of pesticides that are similar in structure to nicotine and aiming to affect the acetylcholine receptors of the nervous system and blocking those. Which leads to paralysis and eventually death for mostly insects. Actually, that was where what they were designed for and what they intended to target. And it was firmly believed that the receptors of vertebrates were much less susceptible for these pesticides. So there's about 7 of them, although there's currently also new ones being developed, and then in different concentrations applied in different substances and brought on the market. But today I'll be mostly talking about, or at least in a meta-analysis, we're talking about 5 different compounds of them, being acetamiprid, clothianidine, imidacloprid, thiamethoxam, and thiacloprid. Imidacloprid is by far the most studied one and extensively used one, but the other ones are definitely also come by in literature, so to say. There we go. So these neonics are known as a systemic insecticide, which means that they are mostly, not solely, applied as seed coatings So they go as they're sprayed basically as a layer, for example, on corn or wheat, with the aim of basically making the entire plant effectively toxic. So all of the parts from root to stem to flower and the seed initially already, they have the toxins and it's spread throughout the plant so that both sucking and chewing insects who would eat the plant would die. However, as we currently also know, whereas it was intended that this would lead to much less leaching into soil or erosion in dust, that was actually not really the case. And it was found out that about 90% actually ends up in the environment. So through most levels of our ecosystems, we now find either residues and metabolites of these pesticides. So that logically leads to the question we want to address here: in what way is that directly, in this sense, affecting birds? So in the case of looking back again to the case of DDT, it was actually found that reproduction was mostly affected in that case. So it led to the thinning of eggshells and thereby a lot of failed hatching and lower reproduction. And in that sense, we wanted to know here what do these neonicotinoids mostly affect? Is that also one part or maybe more, and how does that work? So I basically already said that. The main question to address here are there direct effects of neonics on birds. Indirect effects have been discussed also in literature so far, because of course of the efficient targeting of these neonics that it does in insects leads to a reduced food supply to birds, also believed to contribute to their declines. But here we're therefore focusing on these direct effects. So there's different ways birds can be exposed. I already touched upon that briefly a little bit, but it could be through contaminated water, either through drinking, for example, or even bathing, eating contaminated insects, but also seeds. So where it was nowadays also a bit believed that it was mostly insectivorous birds that could be suffering from these neonics, it might actually also be just a much bigger group of birds. And then assess actually which effects there are. And to basically grasp that concept a bit for ourselves, we made that a bit in this flow diagram. So these effects, of course, kind of interact also a little bit together, where health would affect also behavior, reproduction, and survival, and indirectly also behavior, again, reproduction and survival together, and potentially and a bit logically maybe leading to population trend declines, although we can't look at that within the scope of this meta-analysis itself. And I might be jumping ahead a bit to one of the questions that might follow later. How about modeling this cascade? Because that's something we obviously don't do in the paper later on. We'll come back to that a little bit later. So to assess whether there are effects and also what these effects act upon, we basically divided it into two sub-questions, where we divided or categorized the effects into four main groups, with effects acting on reproduction. So again, this eggshell thinning, survival, so direct mortality or reduced survival in terms of just the days alive. Health factors, so mostly blood values, but for example also physiological parameters like growth in chicks even, or deformities, and behavior. For example, a famous paper from Margaret Eng et al., who assessed migratory behavior after exposure and orientation, for example. Then we also look into these different substances that come around in literature, wanting to find out whether effects that we do observe, whether they actually differ for the different compounds. So is there one that is, for example, one that's much more prone to affect birds in a specific way, yes or no? And we do this quantitatively in our meta-analysis, which is based on 4 pillars, in a sense, based on the PICO framework, it's called, which in our case basically is the foundation on which we developed our search query in our databases. So the population was represented by a bird species, so any bird was included in our study. Any type of neonic that we find was included. And to pass inclusion criteria, the study needed to have a treatment group and a control group. Otherwise, we were not able to use the data. And it needed to be an outcome effect needed to be studied or measured. So one of these 4 groups: reproduction, survival, health, or behavior. We followed the preferred reporting items for systematic reviews and meta-analyses, so a PRISMA scheme. I'll not go into much detail about that here, but after screening in total a little over 300 records, we were left with 50 studies that could be included in our meta-analysis. And to give a bit of an insight, this figure is actually in the supplementary materials of the paper, of how the data in that sense were distributed across the different species, but also which performance measures were studied and which pesticides were studied. You can quite quickly see that domestic chickens, Calis was obviously studied most, because in a sense, most of this research has been performed in the lab. But there were actually also quite some other species that were studied, although mostly in terms of performance, health, and imidacloprid in terms of pesticide. So not all aspects are maybe as represented as you would ideally want, but there's a fair amount of data done onto all these different aspects already. What we then derived from all these separate studies, so we had 50 studies. Most of these studies were actually would include data on, for example, both behavioral outcomes as health outcomes. So we would then derive all these different outcomes measures and turn that into a HEDGE-SG, which is basically an effect size index, allowing you to compare the results of the one study objectively with the results of another. So we, just to summarize that real quick, if you would have the mean outcome of a control group minus the mean outcome of, and subtract that from the mean outcome of the treatment group, and you would divide that by the standard deviation pulled from the two, you would basically kind of objectively get this measure multiplied by a bias measure. So to give a bit of an idea and a feel of how that represents itself with this data, if you were, for example, to have a difference in eggshell thickness of a treatment group versus a control group that was 0.204 by 0.205, you would get a very minimal negative HEDGE-SG. So this was a very small difference, leading although the treatment group obviously had a thinner eggshell, so this is a negative effect that is just very small. Whereas if you would have a very pronounced negative effect in this case of, for example, 50% of the birds dying compared to only 8%, you would get a relatively strong negative effect. So we did this for each of these outcome measures, and then threw those in our models. And I added this here just because I briefly wanted to explain how these models are built up. So we designed multivariate meta-analytical models to try and account for as much of the dependencies that were in this data set. So obviously we have different species. We had similar behavioral or health outcomes assessed within, for example, the same individual. So none of the data, or not everything in the data, is entirely independent, meaning you need to try to correct for that as much as you can, or at least see if that makes for a better model. So we did that in the sense where we started with a very complex model and then compared the models to see actually if those models were even better than just a simple and more elegant model. Yeah, maybe to jump a bit ahead, actually, the phylogenetic effect here, so also the clade the phylogenetic tree that I showed earlier, did not explain or did not improve the model fit, actually, meaning that there were actually very little differences in effects found among the species. So that was actually dropped later from the model as well. And then we tested for each of these. So within the multivariate model, we later added on, that's not shown here in this table, we added the moderators for the performance type, but also these different neonic compounds to test for the differences there. So coming to the results, there's one class missing here, and that will follow on the next slide. That is the survival one. It needed a different effect size because that's a log risk ratio and not a HSG, so it needed to be analyzed separately. But here we have the negative results, or basically all negative results for the other three performance classes. And you can see actually that there was a lot of data, but on basically every aspect that we looked at, we found significant negative effects here, even though behavior wasn't even that extensively researched so far. But the mean, the overall mean is indicated by the solid dot. The bigger bars are the 95% confidence intervals, and since they don't overlap with 0, we assume a significant negative effect here. And we find something very similar here for survival. Most of the effect sizes, I think almost all of them were negative, hence a significant negative effect here as well. Upon exposure to a neonic, there was rarely any survival, actually, regardless of dose, or at least length in days. For the different substances or compounds, the view is slightly different. So thiamethoxam, thiacloprid, and imidacloprid all have significant negative results, but clothianidine and acetamiprid not. But as you might notice, they are relatively underrepresented within this data set, with respectively only 2 and 3 studies performed to them. So that would actually plead for a lot more research even into these compounds still. But since, as some of you might know, some of these compounds are actually already banned, that might not happen anymore anyways. So moving a little bit back, actually, so where DDT was mostly targeting reproduction, we see here actually that it's basically all of these different ones altogether. And then obviously comes the discussion is how does that translate into the effects in the wild, which is an important discussion to have, because most of these studies are conducted into the lab. So it was either that birds were orally fed without having an option so to consume these pesticides, basically. They were force-fed, or eggs were injected in that sense, meaning that it might not be reflecting, in that sense, a very realistic scenario into the wild. However, we ran a subset on analyses also on those studies where birds were just given a diet of these of seeds, for example, with these compounds, which actually yielded very similar results. So in a sense, the complexity of conducting these studies in the field is that difficult that I do feel like the studies which are represented in this data set, give a good view of what would happen upon exposure. Obviously, it's a very tricky comparison to make. But in a way, I think you can argue that even in a lab, you might not get an overestimation of effects, but maybe rather an underestimation. Because if you were to ingest, for example, a seed coated with a pesticide and your orientation or your vigilance is in that matter reduced, you are much more prone for, or yeah, you're suffering a much higher predation pressure in that sense, or are much more likely to get predated. And similar actually in competitiveness, maybe even amongst birds themselves, you are much more likely to get outcompeted or suffer the consequences from just being negatively affected by a compound. Moreover, actually, and I think that's is where we could benefit most from additional research at the moment, is most of these studies, or actually all of these studies that are included in this meta-analysis, are testing for a singular effect of a single compound. And it is actually nowadays almost never the case that we find just a singular pesticide in whatever residue is tested, or whatever groundwater or soil would be tested. These pesticides are always applied in cocktails, and the combination of effects these cocktails would yield, we actually have a very little idea of. So in that sense, the interaction effects here, or the synergetic effects, might actually be much stronger even than what a singular compound would do. And I think there's now 1 or 2 studies that are addressing this for the first time. So it's yeah, that's a very interesting thing actually to now look at. So to quickly summarize, we now, I would say, quantitatively add to the information that we already have on these compounds in showing that there's not only indirect effects, so in the sense of their food availability declining, and therefore population declining, but possibly also just directly, so from not only insectivorous birds being exposed by less food or by eating contaminated food, but also much more broader granivorous birds, or even different diets and other life histories yet again, and also being directly affected. So regardless of diet, there seems to be a risk. And also actually, so I said that already, I'm not sure if everyone was aware, but most of these compounds are nowadays, even beyond the European Union, banned. These bans are reassessed every couple of years. And it's been quite I wouldn't necessarily say a challenge, but these pesticides are continuously being renewed and altered in a way that they can keep being marketed. And this is also known for the neonics. So there's a number of adjustments that have been made, and they have been re-entering the market in different ways. And there's also still parts of the world, the parts also where birds migrate to, actually, where these substances are still used. So in that way, I would say exposure is still there, also because these substances are still found in as metabolites in soil, actually, at the moment.
Alors, Elke Molenaar nous a présenté une étude sur l'effet des néonicotinoïdes sur les oiseaux. En regardant dans le passé, on a surtout en tête la mise en œuvre du DDT après la Deuxième Guerre mondiale et les effets absolument catastrophiques sur notamment les oiseaux. Eh bien, ces pesticides et insecticides ont continué à être développés, et la plus récente variante actuellement en œuvre sont les néonicotinoïdes. Alors, les effets sur les oiseaux sont évidents, mais bien entendu que ces pesticides ont leur effet naturellement sur les arthropodes, mais aussi sur bien d'autres groupes d'organismes. Alors, on connaît 7 molécules différentes de nicotinoïdes, mais il semble que la molécule thiaclopride est soit la plus dangereuse. Alors, des études ont été faites. Ce sont des insecticides systémiques appliqués sur les plantes. Donc, les insectes qui ingurgitent les parties des plantes meurent, mais il faut tenir compte aussi du fait que 90%, 90% des pesticides, du volume des pesticides, continuent à être actifs dans l'environnement, dans les sols et dans l'eau. Et les effets sur les oiseaux peuvent être directs ou indirects. Des études ont été faites, notamment en laboratoire, et l'exposition des oiseaux a été testée notamment par la nourriture, que ce soit des insectes ou même des semences qui ont été contaminées par les insecticides, ou que les oiseaux se baignent ou boivent de l'eau. Les effets peuvent intervenir à plusieurs niveaux, sur la reproduction des oiseaux, sur la survie des individus, sur leur santé, notamment la physiologie, et aussi sur les comportements. On pense qu'il y a peut-être un effet sur l'orientation des oiseaux migrateurs. Et on a étudié les 7 types de molécules. Les populations d'oiseaux ont été étudiés, donc j'ai dit en laboratoire, et on a étudié notamment des passereaux, des pigeons, des canards, donc différentes espèces, différents groupes d'espèces d'oiseaux. Une 50aine d'études ont été réalisées et ont mené à la modélisation des effets. Les effets sur, par exemple, l'épaisseur des coquilles des œufs ont pu être démontrés, et ça a un effet naturellement sur le résultat de la reproduction. Donc la santé, le comportement et la survie ont été étudiés en particulier, et partout des effets significatifs ont pu être mis en évidence pour toutes les molécules, toutes les 7 molécules, mais avec des différences selon les types de molécules. Alors, sur le terrain, il est beaucoup plus difficile de faire ces études parce que les oiseaux rencontrent non seulement des néonicotinoïdes, mais bien d'autres substances qui sont aussi dans l'environnement et qui peuvent avoir un effet ensemble, ce qu'on appelle l'effet cocktail. Alors, les effets direct et indirect, c'est important de le préciser, il y a des effets indirects et indirects. Il y a des effets sur les granivores autant que sur les insectivores. Les risques demeurent malgré des dispositions prises par l'Union européenne, et on continue à développer des variantes d'insecticides, ce qui rend le suivi encore plus difficile. Donc la situation, même si on a réussi à l'étudier avec ce qui est disponible, le développement des insecticides continue à se faire, et il faut le tenir en vue. Voici mon bref résumé. Merci, retour à vous.